Okay, for the third talk of the day, we're going to have Kyogo Kawaguchi from Riken and University of Tokyo.
Hello, good morning. I'm Kyogo Kawaguchi from Japan, Tokyo. So I first want to thank the organizers, Gautam, and everyone for inviting me to this wonderful workshop. It's actually the first time in India for me, and I'm really happy to see this hall and everyone. So we're actually a group of biophysicists. I was just trained as a non-equilibrium physicist, but I've been doing more of biophysics, both theory and experiment. We've been excited about many aspects of many-body physics being kind of like applied or being kind of used in the concepts of biology, how biological systems are trying to like solve many problems using some of these kind of many-bodiness. Originally, I've been working more on the multicellular dynamics, how that kind of relates to active matter physics, or how like interacting particle systems like the Virto model or percolations. And we're kind of moving more towards like inside the cells, what's happening inside the cells, which is probably what we're going to talk about today mainly. And on the other side, I'm also interested in biology in general. So some of that kind of research topic is not even physics. It's kind of related. Kind of weakly related with the chromatin story, but this is more about like morphology or like evolution of the animal shapes. Actually, I was kind of like surprised to see that my former advisor was giving a talk on my like old work at this hall, so you can actually search Masaki Sano's name on YouTube, and you can watch his kind of talk from like 7 years ago if you're interested. Okay, so I'm just going to talk about two works more related to what's happening inside the cells. One topic I'm going to talk about is about the condensates and how we can kind of predict or kind of design some sequences of disordered region proteins and kind of like compare that with molecular dynamics simulations. On the second half, I'm going to switch gears a little bit to talk about the chromatin dynamics. But this is more on the experimental side, and this is a work by Yosuke Bai. He has been taking a lot of time kind of perfecting this method to actually synthesize chromatin and do like biophysics experiments. Okay, so let me just jump in. So you probably heard from last week's Krishna's talk that there's like a lot of excitement going around inside the cells, in terms of the kind of notion that there's like liquid phase-separated condensates inside the cells. And these are kind of an interesting aspect of physics here is that, yes, so cells and biological systems are non-equilibrium, but also the fact that it's like really like multi-component, and it's like really heterotypic. And this kind of aspect is really captured by this problem of this condensate. So inside the cells, there's like typically the order of like several to maybe tens of different condensates that have been identified. And what is really nice is that obviously these are not like, designs and stuff that everyone's kind of like, someone is kind of like putting in the geometry, but this is like self-forming. It's like kind of like a self-organized system in a sense. I just wanted to play this video, which I think is cool. This is like a video I took in our lab. This is like a cell right after cell division. So what's happening is that this kind of red region is the chromatin, and you can see, however, that there's this kind of droplets inside the nucleus, which are the nuclear speckles and the nucleoli. They are the main kind of liquid condensates inside the cells. And you can see that how they're kind of dynamically forming after cell division. So after cell division, the nucleus has to kind of reform its shape, and that's a pretty dynamic process that's happening in the scale of the 20 or 30 minutes. So these are, just by looking at under the microscope, you might have this kind of impression that they're pretty like static and kind of stable, but it's like also like very dynamic and like self-organized in a kind of like a very short time scale. Now, so you probably heard last week, but let me just remind you that this kind of phase separation kind of property is like the one simplistic picture of how we understand these kind of self-organized condensates. And these molecules here in the case is like oil and water, and you assume that the oils have some affinity with each other. And if they kind of like each other compared to water, they will try to kind of bunch up and try to make a kind of a region that is like only made of higher density of oil. And this is like a typical kind of phase diagram that you will see in a textbook as a function of the concentration and the temperature. If you raise the temperature, you would go to a mixed phase. And if you like lower the temperature, we have this phase separated oil and water demixed situation. Now, this critical temperature, in a sense, is kind of like a measure of how this kind of oil droplets or these kind of molecules like each other compared to water. So this can be kind of used as a parameter to kind of characterize the strength of the interactions between the molecules, if you want. Now, so what is the kind of physical mechanism behind this kind of condensate formation? One of the recent interests in this community is on this sequence regions called the intrinsically disordered regions. And these are sequences inside the protein, but they don't really have this kind of 3D nice structures that you can predict by AlphaFold. Actually, to say it in another way, so AlphaFold, you can stick in any sequence you want for the protein sequences, but there are specific regions in your sequences that would not be predicted to fold. And we actually know that AlphaFold is a really good predictor of this kind of like disorderedness. So these are the scores kind of represented by the colors of these kind of sequence. This is a very kind of like very extreme example where most of the region of this sequence is disordered. And it's not necessarily that the AlphaFold is kind of like stupid or making a mistake, but it's really thought that these regions, these kind of orangey regions, don't really have a canonical structure. And these proteins are not really that rare. So if you look across the kind of the genome or the proteome inside the human, so many of the proteins are indeed folded and they don't have these disordered regions, but actually a majority do have some extent of the disordered regions, and actually a pretty non-negligible amount of them actually are like highly disordered, meaning that maybe like 80 or 90% of them are these kind of disordered region sequences. And also these disordered region sequences are not just random sequences. It's like a really easy test you can do is you just take just a random region of your kind of IDR disordered region sequence, and you stick it to GFP, which is a fluorescent protein, and see how they kind of localize inside a cell. And typically they do have some kind of localized kind of signal, which means that this kind of really short kind of sequence of amino acids also already has some kind of information regarding localization, which is kind of presumably due to some physical interaction with other proteins or RNAs. Okay, so these kind of disordered region sequences, they're still proteins, so we know what they are made of. So there's no mystery behind the interactions between these amino acid sequences. And so now we have a pretty good idea of what are the most kind of like dominant or important interactions that would drive these condensates. And these are typical examples of the four main kind of drivers: the charge-charge interactions, cation-pi interactions, dipole-dipole interactions. And recently, people have appreciated more of the pi-pi stacking, which is a little bit has been kind of not being too kind of focused in the kind of folded protein interaction community. But now people are realizing this is very important in the disordered region sequence interactions. So, so this is one of the questions that we wanted to pose when we started this project. Is that so? We have these many droplets inside the cells, and they are coexisting. So now that is somewhat of a problem because these droplets have to coincide while these molecules are being very dynamic. So as I want to remind you, so these liquid condensates are not these kind of static aggregates, but rather the molecules are kind of like moving around and diffusive, and they're pretty rapid. So even if you see under the microscope that these kind of droplets look quite static, the actual molecules themselves are moving around, and sometimes they will just jump out from the condensate, and they'll try to come back, and not necessarily in the same condensate, but they might diffuse to another condensate. But even in that kind of like dynamic situation, so these molecules somewhat kind of like remember their identity, right? So if it's coming from a red condensate, they would never jump into a green condensate, and vice versa. The green molecules would never jump into a red one. So the reason behind that is simply that, so these kind of red molecules, they have an affinity with each other, and the green molecules would have affinity with each other, but the green and the red ones have some repulsive interaction. So they have to basically hate each other. They have to hate like themselves, each other as much as like water. So this becomes a problem if you want to have like multiple condensates kind of coexisting, because I mean, designing this kind of affinity is pretty simple, but you also have to guarantee that these kind of like new condensates, each time you kind of like make these droplets, you have to make them repulsive. So you have to make sure that these kind of attractive interactions, if you introduce them, they have to be, in a sense, like orthogonal to the ones that are already existing. And that seems quite implausible if you think of that like limited kind of set of force fields that you're allowed to use in the amino acid sequences. And I mean, the short answer is that yes, so we actually do. If we kind of ask this question, yes, we don't really have that much of a space if we just only think of the amino acid interactions. So to kind of answer that kind of question, what the path we took is to do some kind of theory. So ideally, we want to kind of connect this path, right? So if you're given some disordered region sequence, amino acid sequence, then you want to predict whether the sequence would phase separate inside a cell, or would they like demix? So it's known that certain condensates, the green one or the magenta one, they have different types of sequences. If you introduce a new sequence, which one would it go to? Or if you had like two sequences, would they separate into two, or would they kind of come into a same droplet? So that's the kind of the question that we want to answer, like given the function or the kind of the input of the sequence. But unfortunately, the experiment, we don't have a rich enough data set yet. We have some nice in vitro experiments, but that's also somewhat limited to some specific proteins. So what people are now actually doing is to do the coarse-grained simulations of these sequences. So the idea here is that you want to simulate a pretty large scale. So you inevitably have to coarse-grain your proteins. So you just take the sequences and you coarse-grain it so that it's like a single residual level. So it's like a chain of amino acids. And each amino acid is just going to be like 1 degree of freedom with 3D moving directions. And they're going to be chains. So they're going to be connected by a spring. And they're going to have like specific interactions corresponding to each amino acid type. But that's the extent. And you think that the water is also a Langevin, basically. It's just it's like a noise field. It's not like just try to like make the degree of freedom as small as possible and still capture the sequence dependence. And for that kind of sequence, people have succeeded in like making these kind of phase separations realized in silico. And also they can now compare that with the experiments or the in vitro experiments. So they have like a nice parameter set that is thought to kind of like recapitulate the real experiments. Okay, so then the question is that, okay, so it's pretty difficult to do this thing. So the theory connecting this sequence to the experiment. But at least we should be able to do this. So we have a given sequence and we have the given kind of force field. Can we at least predict the macroscopic structures of the MD simulation? So this seems like a pretty well-defined stat-mech soft matter problem. So this is the one that we tackled. Okay, so just a small note on the relation to this kind of macroscopic theory. You might think that this is just a multi-component Flory-Huggins theory. So why don't you use that? So now the question that we're basically posing is that, okay, so we in principle, if we have this, yes, we may be able to get the phase diagram. But how do we know this kind of chi parameter? This chi parameter is the interactions between the different sequences. And we basically want to know how we can get from the microscopic parameters to get this kind of macroscopic interaction between the sequences. And there has been like many attempts, so we are not the only ones doing this kind of question. Just as a reference, we have this kind of case where there's only charge, this thing called the random phase approximation, originally coming from the condensed matter community. This has been quite successful in kind of predicting the mutation kind of experiments from specific sequences. And also, I think Krishna talked about this kind of random matrix approximation. This has some implications about what could be going on inside cells. And also, it's kind of like already like a notion that if you try to do some kind of prediction between these kind of polymer sequences, you kind of run into the quick problem that at the amino acid scale, the rank is pretty low already. So you already kind of have an idea that the chi is probably going to be low rank, but the question is how the polymerness would kind of like change that picture potentially. And also this kind of the other kind of like a little bit more specific problem to this community is that, okay, so it's also known that even if you're given all the parameters of this kind of free energy Flory-Huggins, it's still difficult to draw a phase diagram if it becomes like multidimensional. So if you have like more than 3 components, this becomes like very, very difficult, and is like dedicated kind of methods or computational methods that have been proposed specifically for that problem. So another kind of like question that we want to ask and to answer is like a workaround. I mean, can we avoid solving this whole thing and try to say something meaningful about the sequence interaction affinity or the repulsiveness? So the kind of like the stat-mechie approach that we will take is that we're going to assume that, okay, so from the kind of expansion from the low concentration, we can get this free energy, and that would kind of define this kind of 2-body interaction parameter, which is in this case the virial coefficient. And this B is going to be represented by something like this. And this B is like this. It's basically the effective interaction between two sequences. And this U is going to kind of include all of the interactions between the polymer. So in principle, if you can kind of solve this thing based on all the interactions between each amino acid, just solving this kind of two-body kind of integral problem, then you should be able to get this B. So the VR coefficient has a physical kind of interpretation as this kind of the volume of this amino acid, meaning that it's like effectively becoming zero when there's like no interaction, but it's going to be like positive or negative depending on the kind of effective repulsiveness or the effective kind of affinity. And this U is going to be this V is going to be a function of temperature. So just by changing the temperature, you would assume that the two-body interaction would become cross over from affinity to a repulsive interaction. Okay, so now what we actually try to do is to estimate this BT in a simpler way. So if you think about really doing this calculation, you have to integrate all the kind of single amino acids, which is somewhat hopeless. You can do it numerically, but you don't have a kind of like a good analytical way to do it. So what we're basically proposing is to do approximation for that part. So how we do that is that, so this is the most crude way that you can think of, is that you're just going to break up all the amino acids into single bits, and then you just kind of like calculate the interactions of the individual amino acids, and then just like add them all together. So this is what we're calling the monomer pair approximation, which is kind of like a coarse-grained, kind of like a, oh yes? Yes? Yeah. Yes. Exactly, yeah. So each chain has its ring, yeah, yeah. So yes, so this is, in that respect, this is like forgetting all about the spring. And this is like treating each amino acid from each two sequences by pairs and just summing them up. This would basically end up, it's a really simple calculation. So you just need to do integrals across the 20 by 20 kind of interactions. And if you get this matrix, which is going to be represented by this VMP, then afterwards, if you want to calculate the sequence dependence or the interactions between two molecules, all you need to do is to sum them up. So you just need to count the number of the amino acids from one sequence and count the number of amino acids from the other sequence and just sum them up using this kind of linear kind of combination. So obviously this has a really kind of problem. So it captures the number of amino acids inside each sequence, but it forgets about the sequence dependence. So it's not really kind of taking into account which amino acid came after which amino acid, etc. So to kind of like take that into account in the lowest kind of lowest expansion in a way, is this kind of dimer pair approximation that we're proposing. And this is very similar, except that now we're treating dimers as a monomer. So we're going to take two sequences that are like two amino acids that are kind of adjacent to each other. And then we just take the integral of these two dimer pairs and then try to sum that up. So there's actually no formal way to kind of derive this from first principles, but this is like a heuristic way that we kind of cooked up. And then we just sum this up. And as I said, this has both the composition and the sequence dependence. So the order of the amino acid somewhat matters in this approximation. Okay, so what we have to do is to run many kind of coarse-grained simulations for ourselves. And we just take the sequences of the disordered region sequences from the human proteome. And we test and see the kind of the temperature-dependent kind of phase separation properties. And later the two different sequence interactions, but I'll come back to that later. But I mean, basically the order of the database, like maybe like 200 to 300 distinct sequences, and we just restricting to a 50-mer disordered region sequences. We have some bunch of like ways to predict the critical temperatures from this kind of limited number of simulations, but that's also in the detail. Actually using this Genesis, which is one implementation of this kind of coarse-grained sequence, coarse-grained simulation. Okay, so this is the first result. So we just calculate this interaction. As I said, we want to predict the BL coefficient. So one kind of like simplest kind of test would be to see directly compare this kind of BL coefficient, the real BL coefficient versus the predicted. And the way to do that is to see the crossover point of this one to 0. So that's called the Goyal temperature. And that temperature would be predicted. So if this is predicted nicely, then you should have a correspondence between the prediction and the real calculation. So for the real ground truth that we're calling, or in a sense experiment, is also a numerical simulation that we're doing. And we can compare that with that integral formula that I proposed. And this x-axis is that simulation or the experiment that we're calling. And the y-axis is the prediction. And this is the monomer pair approximation. And you can see that it's like already doing pretty well. It's kind of capturing the kind of like the difference across different sequences. But you can see that it's not capturing the sequence dependence, obviously. So here what I'm doing is that I'm taking some sequence, and then I'm kind of sorting the amino acids in alphabetical order. There's no meaning to that order, but that would just generate many of these kind of blocks of these amino acids. And that would, you can assume that that would change the critical temperature or the Goyal temperature. And it actually does change it a lot. And if you also shuffle it, that would also change it a lot. And those kind of the differences will never be captured by this kind of monomer pair approximation. Now, if you go to the dimer pair approximation, that part improves quite a bit. Although sometimes for the sorted sequences, it would come off, but it's not really a systematic kind of like difference. So this is pretty good. And now we directly test the TC, the critical temperature. So this is based on the phase separation simulation. And this is pretty good. So we compare the simulation TC versus the theory predicted critical temperature, and they are nicely aligning. So here, I'm actually changing the different force fields. So there's no kind of consensus of what kind of parameters or the models that we should use for this coarse-grained simulations. There's basically two kind of schools of thought. One is called the HPS, which is taking into account it's kind of adding on top of the Leonard-Jones potential a kind of hydropathy score, so how they kind of basically hate the water at each amino acid level. And that has been used, and the parameter has been tuned by different groups. And we used two versions of that. And we also took the MPy model, which is another different force field. It's not using the Leonard-Jones, but it's a different force field. And they have more emphasis on this kind of pi-pi interaction that I talked about. And these are like quite distinct parameter sets. And you can see that the prediction of the temperatures are quite different across the different force fields. But the claim that we're basically showing here is that for given force field, we can predict what's going to happen if you do run the simulation. So that's just based on that integral that I showed you. Okay, so now we move on to what happens if you have like multiple sequences. So here we have like 200 protein sequences of different sequences. Sorry, you have basically, so we have two different sequences, and we have 200 each of them. And then just run the simulation. You can see here that it's like demixed. So the green sequence and the magenta sequences, they both want to phase separate. So they are doing this kind of condensate thing. But you can see that they're not really mixing nicely. Now, if we run this simulation for many different kind of combinations of these sequences, you see that it really depends on the sequence what would happen. And that example is depicted here, the movie that I showed you. So in these two specific sequences, they would basically demix, and they would not kind of like coming together. But there are cases where it looks more mixed or like random. So here, you don't really have much of a discrimination between the spatial positions, distributions between the magenta and the green sequences. But you can also have like a hypermixed state that we're calling, which is saying that the green ones and the magenta ones would basically like each other than themselves, basically. So this kind of like different, having this kind of pair sequence would basically help them phase separate more. Now, using that kind of framework that I proposed, using that integral thing, you can basically also calculate like the effective metric of the repulsiveness. And what we found is that this kind of really simple formula, we just take that kind of high parameter, which we can directly calculate from that BRL coefficient. Then we just like, in a sense, we kind of take the diagonal part and just focus on the off-diagonal part. And then we can actually have this nice correlation between that value and the phase separated so the demixing kind of feature. So for the demixed pairs, this value tends to be positive. And for the more neutral ones, this value also tends to be close to 0. And if it's negative, it's going to be like this kind of hypermix. So we can kind of more or less predict before doing the simulation what's going to happen if you mix in two different sequences. Now, so we can use that to design or kind of like see which kind of pairs would basically demix, and we can use that and apply it to multiple different types of sequences. So this is an example where we take like 3 different sequences and see if they would really phase separate, which is predicted by the theory. And this is what happens. So if you just take these 3 combinations, by the way, these are really taken from the human proteome sequences, and they would actually have these kind of nice 3 layers. I wanted to extend this and go to 4 sequences, and we couldn't really find a good combination. And this is obviously because one reason could be that the human proteome IDR sequence is not as diverse as we want it to be. But maybe we thought that there could be some theoretical bound too that could be contributing to this kind of limitness. To see that, we can kind of like go a little bit further with that theory. Some simplification here, but let me just like go through. So here, the kind of effective interaction that I introduced briefly can be now written in this kind of form. And here, this VMP, if you remember, is the effective interaction between each single amino acid. So this is just a 20 by 20 matrix. And this N is the composition vector, which is just basically counting how many amino acids are in this each sequence. And this is what it's saying is that if you want to compare 2 sequences, I and J, you just take these composition vectors and take the difference, and you just like basically calculate the square of that with this V matrix in the middle. And so this is like a linear algebra problem. So here, this value is important because from this you can predict whether the pair is going to be more attractive versus repulsive. So the kind of the sign of this chi is what we want to predict. And that is basically, you can think that this is basically predictable by the eigenspectrum of this V. Now, if you look inside what's the eigenspectrum of V, then it has a very kind of low rank structure, as was predicted from the old work by Wingren. And this is saying that, so there's like very specific kind of eigen vectors, the directions in your amino acid composition vector that would lead you to this kind of more demixed state. And also very specific directions which would lead you to a very strongly mixed state. So if you just look into what's happening in these kind of like more demixing vectors, they actually kind of like correspond to exchange the charged amino acids to the aromatic residues, or you just change these kind of strongly interacting ones with the other sequences. So that's like the kind of insight that we can get. And these kind of 2 negative eigenvalues can be kind of combined to generate many demixing condensates in principle. I ran out of time, so maybe I can skip a little bit, but this is kind of like a nice kind of geometry problem that you can map to. So you can think that in this kind of 2-dimensional plane, which is basically here, you only have 2 dimensions that you're allowed to demix into. So you can kind of reduce the problem into 2D. And in this kind of 2-dimensional kind of manifold, if you want a little plane, you try to like position your sequence, and each sequence is going to correspond to a single point in this kind of 2D space. And the problem of finding 2 demixing condensates would correspond to having 2 points that are far apart enough in this plane. And you can kind of think that this is somewhat restricted because you don't have like an infinite plane in this 2D. So you're only allowed to kind of find your sequence within a certain kind of region. So that would kind of make your kind of like a choice quite limited. And from here, you can kind of deduce that the maximum amount of kind of demixable sequences will be maybe like 3 or 4. So this is all like given the kind of force fields that I said, but this we tested for the different force fields that are used in the community, and for all of the combinations, this was basically the same. Now, so the kind of the takeaway from this work is that, okay, so these cells can only phase separate maybe 3 or 4 condensates, which is pretty bad news. And if you're only using the disordered region interactions, that's in a sense like a limit. And I guess we get a lot of questions, what would happen if we introduce RNAs? And that's a really interesting and important question. But the short answer is that the RNAs are just like really negatively charged polymers. So just by introducing them would not really change the picture a whole lot. What is actually really probably happening inside the cell is that the RNAs have very specific interactions with the proteins. So if you kind of like introduce more of these kind of like not really simple polymer interactions, but rather really specific sequence dependent interactions, then you might be able to kind of, in a sense, make your dimensions higher. And also there's like orthogonal strategies that the cells can be taking, and that's also really interesting problems. And also there's kind of shutting out these kind of words in case that people are interested. I'll be happy to discuss more. But recently, people have been interested in the microphase separation, trying to predict that, but clearly that's very difficult. But that's probably going to be more important in understanding these biological condensates. Okay, there's a question? Yeah, sorry.
About the RNA, so what do you think is it that's going to make it more complicated? Is it also the geometry of the RNA, like the geometry of the structure, or what?
Yes, exactly. So the RNA's sequences themselves could be read by the proteins, and this could be through the sequence themselves, but also their the folded structure themselves. But yes, so that kind of folded structure would not be just like a simple kind of simplified polymer interaction, but rather that folded structure would be specifically targeted by a certain protein, right? So that's in a sense, that's kind of like a totally different dimension that you want to include. Okay, any other questions? If not, in the remaining time, I want to jump into another topic. So the other very important heteropolymer inside our cells is the chromatin. So chromatin is something that I totally neglected when I showed this picture previously, but this red part, that's the chromatin. Specifically, that was actually staining the histones. So these histones are the proteins that are wrapping around these DNAs. And these proteins are kind of having these kind of like epigenetic marks, so-called, which are like methylations or acetylations. And depending on these kind of marks, it's known that these kind of different regions in your chromatin would have different kind of structures, or these kind of 3D structures. And that would probably lead to different accessibility. So the more open regions are probably the regions that are like more open in the sense of the gene expression themselves. So the open regions are what's called the euchromatin from more than 100 years ago, and the more closed regions correspond to the heterochromatin. Now, as I said, this is like somewhat more interesting and important if you think of in the context of the gene expression or the kind of like cell type specificity. So this is kind of related to the previous talk as well. But if you look at the whole kind of like geometry of the different cell types in our body, the cells basically have the same genome, but they have different gene expression levels. And inside the dynamics or the kind of the underlying kind of variable here, one of them will be this kind of like chromatin structure. So there's like a lot of biophysical and biological interests going on in trying to understand the 3D structure of these chromatin structures. And there's this kind of really interesting development from like maybe 15 years ago that people are now using next generation sequencing technology to try to deduce the 3D structure of the chromatin. And this is called Hi-C. And this is basically a way to get the contact map of the chromatin. So the idea is that inside the cell, you cross-link the proteins. And this would lead basically to freeze the 3D structure. And then you chop up the DNA. And then you do something called the pair-end ligation, basically connecting the ends that are becoming free because of that kind of breaking up. And that would lead to these kind of like artificial connections that are not the real connections in the genome, but rather just based on the proximity, just by chance, the sequence regions that are close together would come together in your data. And just by reading these kind of accounts of these kind of paired regions, then you can get the so-called contact map. And here, the x-axis and the y-axis both correspond to basically the address of on your chromatin, the positions on your chromatin sequence. And this kind of redness here corresponds to how many of these kind of pairs of the two different sequences you found in this really big data. So this kind of count, you can naively think of as kind of like kind of recapitulating the 3D structure. So if you're given a 3D structure, you have this proximity map represented by this kind of Hi-C data. Now, from this, people have now like some kind of vision about what could be happening inside the cells. The biggest structure that we could call is called the AB compartments. And there's like more smaller structures which are called like topologically associated domains. And there's like more smaller scales called the loops and etc. But now the interest here is that basically you can kind of compare this kind of like model with some polymer physics as well. Now, the other interesting thing is that these kind of compartments of the 3D structure, they actually change according to cell differentiation. So if you look at different cell types, they have pretty different structures in the 3D chromatin. So people are kind of like thinking that this kind of change in the chromatin structure is somewhat related to the robustness of the cell fate or the differentiation. Okay, so given that kind of background, we are kind of taking this biophysics or in vitro approach that we call. So there's like lots of different components that could potentially be leading to the difference in the 3D structures. And there's kind of specific labs targeting each of these very interesting epigenetic structures, or even recently like non-equilibriumness, because those kind of structures are maintained also by these kind of the motor proteins called the cohesions or the condensins. So that's a really interesting hot topic as well. But now we want to take a step back and try to think about from the first principles polymer physics, can we compare what would happen if you have a really large chromatin structure, chromatin sequence, and we just put it inside water? What would the structure be, or what the dynamics would be given that material? So here we want to take this kind of in vitro one-by-one approach, which is what the molecular motors people did maybe like 30 years ago. So to do that, we're actually really making chromatin. And there's actually a way to do that, which is pretty well established. You just purify the histone octamers, these protein octamers, and you mix it with the DNA, and you do something called the salt gradient dialysis, which basically change your interaction between the DNA and the protein in a very kind of mild and slow way. And that would lead to this kind of nice ordered structure. And you can actually use these kind of really nice sequences of the DNAs, which has been characterized, and you can kind of even position the histones on which positions they would come into. So you can basically target and make a certain like 100 mer chromatin in this kind of old method. The problem about this method is that you can't really pre-pattern the positions of the epigenetic marks. So people have tried to go around that by basically doing the kind of this salt gradient dialysis thing, the reconstitution first, and then doing the ligation. So you just make these small bits, like in like a one nucleosome, and you have different nucleosomes with different epigenetic marks, and you ligate them or connect them in the order that you've designed them, and then you can get different sequences with different kind of epigenetic marks. In this case, they had the tails. The histones have these kind of things called the tails, which are kind of epigenetically modified. And it's known that if you have this tail or not would really matter in the interactions. In this specific work, they want to test that interaction kind of difference across the different types of polymer configurations. So what we actually take from this work is to just make it much bigger and just make an array of 12 mers, and then we just make 12 mers each reconstituted in different tubes. And then we just put them together and make like a really large 96 mer structure. So 96 mer is about like 20 kb in the base pairs, and this is about the single gene kind of length scale. So that's what we are trying to target. Okay, so this is the result of what we are getting recently. So this is the atomic force microscopy. You can see actually that the nucleosomes are nicely forming, and you can actually count the number of the dots. And because Yosuke put a lot of work in kind of perfecting this kind of dialysis method, you can actually count, it's like really close to also almost perfect. The problem here is that what's really kind of biochemically challenging here is to do this kind of ligation after these kind of reconstitution. So biochemically, it's much, much easier to do the ligation first. That's because of the concentration of the magnesium. But that would basically, it's like a very narrow window that Yosuke found that would allow you to do this, but still the dialysis, the ligation is not perfect. So you're not really getting 100% of these 96 mers, but you're getting like something in between as well. You can kind of take advantage of that and try to find that in the AF. Oh, this is like one example. You can actually see that it's a real intact one polymer, and you can actually count the number of integers. And you can actually see the different lengths of the sequences, and you can actually go in and count them one by one, and you can see that they're also very efficient. So it's really the kind of multiples of the 12 mers are really realized in this setup. Now, using this, what we want to do is to characterize the dynamics. So this is you tether one end of this chromatin, and you just look at what the fluctuation is going to be in the other end. And you can see here that the yellow dot is the dye at this kind of open end, and the red dot is the tethered end at the bottom. And you can see that the red one is not really moving, but the yellow one's really moving quite rapidly. And you can also apply force or the flow to this situation to see that this is a real kind of chromatin that would match in the sense of the length. So after that kind of post-filtering of the data, we can see that this fluctuation is coming from a real 96 mer full-length sequence. So you can see that by changing the salt concentration, and also we can see that changing the sequence, epigenetic mark pattern, that kind of actually dynamics and the length scale actually changes quite rapidly. So one prediction that we would have is that if you acetylate all the histones, that would lead to more repulsive interactions between the monomers, which would make your chromatin more open. And this will make sense because the acetylation marks are mostly found in these kind of open regions of your chromatin. And if you don't have that acetylation mark, you'll have like a much smaller structure. And you can also measure the dynamics from this movie. And if you compare the kind of dynamical kind of time scale of that fluctuation with the kind of length scale, the size scale of that fluctuation, then you can compare that with the polymer physics prediction. So now one model that we found is most fitting is the so-called ZIM model, which is thinking of a kind of interaction-free polymer, but you add on hydrodynamic interactions. So this specific model would lead to prediction that the relaxation timescale and the fluctuation length scale would be kind of like a cube relation. So this is a kind of proportional relation. And if you look at this kind of like coefficient here, you can calculate by doing lots of integrals, and that actually pretty well matches with the experimental observation. So we think this is probably the right one to model this kind of length of the chromatin dynamics. So just for the last part, sorry, I'm running out of time. But here we actually now have done this kind of high-C experiment, that contact map experiment in vitro inside the tube. And this was very, very difficult, but we figured it out quite recently. Now we do it inside the tube. So this is for the in vitro sequences with 12 mers. You can see that they are like high contact in between the intramolecules, and across molecules, you have no signal. So that's like a benchmark, it's working. And then we apply that for the 96-mer, the large structures. And here the 96-mers have different blocks of the designed modifications. So these blue regions correspond to the unmodified histones, and the red regions correspond to the acetylated histones. And you can see that depending on the design of your chromatin, the 3D contact map structure is quite significantly different. And for specifically this kind of like zigzag kind of pattern, you do see this kind of like nice checkered board kind of pattern, which is predicted by theory as well. One very like small remark, if you're kind of like interested in this kind of high-C contact map, if you're in this community, we also measure the scaling of the contact. And we found that surprisingly, all of the data suggests that the power is like minus 1.5, which means that they're really kind of Gaussian chains. And that actually doesn't really make sense. So we think that that's probably some kind of methodological kind of artifact. But that's probably not just our problem, because if we look into the literature and the other data, which have also achieved a very high resolution contact map experiment using the real cells, in the same kind of like regime or the range of the distance, you actually see the same kind of parallel. So my take here is that there's probably some reason that in this kind of length scale, still short compared to the whole genome or the chromosome scale, there might be some reason that this kind of high-C method can only produce this kind of parallel, but we don't really know the physics behind this yet. Okay, so that's about it. So it's like a method development thing, and we have a bunch of kind of validation methods that we had to invent along the way. And as the findings, what we're excited about is this kind of single polymer fluctuation, which for the first time has been compared with the ZIM model. And we can actually artificially make the structures, which would probably lead to the understanding of the biophysics of those real 3D structures on the chromatin. Okay, so I'll probably stop. So the first part of the talk was about Kyosuke's work on the heteropolymer interactions. That was a theoretical work with numerical computation. And the second work was about the chromatin reconstitution that was led by Yosuke Fukai, who's a very talented postdoc. But many of our colleagues in our group, the technical staff members, have been helping us a lot. And we also have really nice collaborators who are really specialists in chromatin reconstitution and biochemistry, specifically Kuromizaka Lab, which is one of the leading labs in the world in this field, and Umehara Lab, who's specific in specialist in making acetylated chromatin. So they actually provided the materials for us. So in our lab, we are doing like a pretty broad range of like biophysics problems. But now we have a new lab in the University of Tokyo, and the RIKEN lab is actually moving to Tokyo next year. So I'd be happy to take questions and discuss the other points that I didn't really have time to discuss today. Thank you.
about the first.
Sure.
I had a question about the first part. In the end, you had this two-dimensional box and you fill in 3 or 4 blocks.
Yeah. Oh, yes. It kind of does, but kind of doesn't. So the size dependence would appear as a scale. So that would actually change the, so here. Oops. Yeah, so here, the biggest thing that would change is the TC. So if we just take the same sequence and double it, the interaction property will be very similar, but the amplitude will be just stronger. So that would shift the TC. So if you, this is just constraining on the single length scale, but if you just go, just like do 100 mer, if you do the same calculation, with the same geometry, but you just have the ICTC circle all changing. So this is just showing like a shorter, like a very low temperature, but this is because it's 50 mer. If you go to 100 mer, it'll be more realistic and go to 300. But basically the sequence-specific interaction picture is the same. It's just the scale. Yeah.
I was just curious, like you showed the relaxation time calculation using the ZIM model.
Yeah.
Can you talk more about the length scale of the polymer? Because I was just wondering whether larger modes of Rouse model will replicate the same results. So did you?
Right. So the distance dependence actually would not be. So in the Rouse model, without the hydrodynamic interaction, would be square.
Yeah.
So that's the difference.
Yeah. So I was just wondering whether, like, what's the length scale of the polymers you are using, where hydrodynamic interactions become important here. So, like, can you talk about the length scales of the polymers you are using? Like,
Oh, I see. So here, what we're basically saying is that the length scale changing depending on the epigenetic patterning,
okay.
and the actual length is the same, 96 mers.
Okay.
We're basically saying that the effective length scale changes according to the different, epigenetic patterns. And yeah, this is saying that that changes, and that change versus the time scale is, cube. Yeah. Oh, yes. Shuffling was random.
So what was the point that you were trying to make in that plot? Like, I think you were trying to say something like claiming why dimers kind of interaction will help you to compute the
Yeah.
So here, why are the points of all sorted and shuffled lying together in the dimer pair approximation? Can you talk about it a bit?
Yeah. So here the simulation data is the same for the two plots. Yeah, the x-axis is the same for the two plots. The y-axis is depending on the different models you predict. And what I'm here saying is that this kind of sorted sequence, this red dot, probably has its pair somewhere here, which is the original sequence. And by sorting or shuffling, you would basically shift only in the simulation direction. But by definition, the monomer pair approximation, the y signal, the y-axis would not change. But in the bottom, that part would also change. So that's why, so here you can see clearly the red ones are totally off, right? But here the red ones are still somewhat off, but they are still kind of close to the line. That's what it. Yeah. Exactly, yeah. I'm not sure if I'm answering your question, but just empirically, what we do find is that shuffling tends to make the simulation value a little bit smaller, but that's not as big as when you sort it. So the sorting is a pretty big perturbation. The shuffling is much smaller, but the claim is that that is still kind of like somewhat better. The yellow cloud is a little bit better here too, right? Yeah. So if you, this kind of Oh, this was, yeah, it does have a Pearson. This doesn't have a number, sorry. It's in the paper, but the Pearson correlation using all the data is much better if you compare this one to that. Is that answering your? Sure, yeah. There's no.
Any other questions? Okay.
Yeah, then maybe I can, sorry, I'll talk a little bit about why I just have this kind of hidden slide. So just empirically, what we do find is that, oh, is what happened? Oh, sorry. Well, maybe it was in the middle. Oh yeah. So this is sorry, it doesn't really have much explanation, but these kind of x-axes are the things that we know would screw up the predictions. So this kind of method is an approximation, and we know the regions that would fail. And if you basically have lots of charged residues, it would tend to fail. And if you have like lots and lots of aromatic residues, that would tend to fail. And the STD is like looking at the kind of blockiness of these kind of charged domains. So if you have lots of like positive charged regions in the sequence, that would make this value also be large or small. And if you go like extend these kind of values, then for those sequences, you would tend to have like a larger error. So which means that mostly the kind of natural sequences are not that extreme. So these are the distributions of the sequence that we used in our calculation, the white one. And this blue IDRM is the whole human proteome. And in all cases, I mean, the dominant majority of those regions are within the region where the simulation basically works. So yeah, in a sense, one way to understand our approximation is that it's not like a universal workaround, but it's like a kind of like an approximation method that works at least within the range of the real sequences. I don't know if that maybe just confused people. Okay, yeah, I'll be sticking around for this week, so be happy to discuss more.